Welcome all to this Psychiatry CPFT seminar. I'm Saurabh, postdoc with Valerie Woon. Today I'll be filling in for her. She's told me she might be slightly late and join very soon, and she sends her apologies for that. So today we have an excellent speaker with fantastic clinical work by Dr. Nicole. I'd like to introduce Dr. Nicole Provenza, who is a postdoctoral fellow in Dr. Samir Sheth's laboratory at the Baylor College of Medicine in Houston. She completed her PhD in biomedical engineering in David Burton's lab in Brown University in 2021. And she was awarded the Draper Fellowship for her graduate research. Her graduate research focused on developing adaptive deep brain stimulation for obsessive compulsive disorders. And as part of her graduate work, she developed a research platform to collect intracranial local field potentials, synchronized with disease-relevant behavior at home to aid in the identification of neural biomarkers of obsessive compulsive disorders, symptoms in natural environments. And today she'll be speaking on the similar topic about candidate neural biomarkers of OCD symptom intensity and response to deep brain stimulation. So I hand over to you, Dr. Nicole. Please go ahead.
Thank you so much for the introduction. I'm very excited to be here. It's early in the morning for me, but good afternoon to you all. So I'll just get started. So the overarching goal of my work is to improve the efficacy of deep brain stimulation for treatment-resistant OCD. As you all know, OCD is a psychiatric illness that is sometimes debilitating and marked by obsessions, which are recurrent distressing thoughts, and compulsions, which are repetitive ritualistic behaviors. OCD affects 2 to 3% of the US population, and first-line treatments include medication, namely serotonin reuptake inhibitors and psychotherapy. 10 to 20% of OCD patients are treatment resistant. And we know that DBS targeted to the ventral capsule ventral striatum offers relief in 50 to 70% of refractory cases. This target, the ventral capsule ventral striatum, was introduced originally based on the target ablated in classic capsulotomy procedures. And over time, the target was refined to focus on the ventral anterior limb of the internal capsule and nucleus accumbens. And so this target together is now referred to as the VCVS. And VCVS DBS is approved in the US as a humanitarian device exemption by the FDA. So one of the reasons that DBS might not be 100% effective today is that DBS amplitude is constant, but symptoms are not constant. So if stimulation amplitude is set too low, you might still see OCD symptoms. But if DBS amplitude is set too high, you might start to see side effects of VCVS DBS, the most prominent of which is hypomania. So getting impulsive, decreased need for sleep. So during a DBS programming session, the goal is to find the stimulation amplitude that reduces symptoms without causing side effects. So it's kind of like a sweet spot. But the problem is the sweet spot can change over time, especially after initial programming while parameters are still being optimized. So one of the challenges with successful programming is finding this sweet spot. And the current paradigm of DBS today is open loop, which requires several visits to the clinic after DBS is turned on for the first time to optimize stimulation parameters. So the patient comes into the clinic, DBS is turned on or adjusted. They might say, I think I feel better, but then they go home. They might experience side effects, hypomania, depression, sleep disturbance, or even new OCD symptoms. And weeks, months later, they go back to the clinic and they don't feel better anymore. So tonic stimulation really fails to address the dynamic nature of OCD symptoms and VCVS, DBS side effects that might fluctuate over minutes to days in between clinical visits. So we're proposing that automatic detection of mental states related to symptoms and side effects might improve efficacy of deep brain stimulation for OCD. So what that would look like is we would continuously record neural signals from the brain, use those signals to classify brain state, such as an obsessive compulsive symptom state or a hypomanic state. and then use that information to control stimulation parameters to increase or decrease stimulation amplitude accordingly and relieve symptoms. So there are many, many key challenges that need to be addressed in order to get here. The first two I'll note involve better characterizing symptom state. So there's a lack of established physical readouts of symptom state in OCD and other psychiatric disorders. And there's a lot of uncertainty in how symptoms are varying over time at home. And the last involves neural biomarker identification. So today there are no robust neural biomarkers of any psychiatric disorder. And until recently, we didn't have the ability to record intracranial neural data at home. And home is really where these symptom fluctuations and side effect fluctuations are taking place. For example, onset of hypomania takes days to weeks after stimulation amplitude is increased in the clinic. So we think that capturing neural activity at home might be a more fruitful path toward biomarker identification rather than simply capturing it in the clinic at these time points that are kind of spread far apart. So the work I'll present today is published in our paper from December 2021 in Nature Medicine. And this work was part of a collaborative NIH-UH3 effort between Baylor College of Medicine, led by Wayne Goodman and Samir Sheth, University of Pittsburgh and Brown University. And the overarching goal is to develop adaptive deep brain stimulation for OCD. And we really try to tackle all the key challenges that I've listed. Most notably, the ability to detect neural biomarkers outside of a controlled clinical setting. So in this work, I'll talk about 3 of our first 5 study participants, who I'll refer to as P1 through P5. 2 were implanted with the Medtronic Activa PC plus S, and 3 were implanted with the Medtronic Summit RC plus S. Both of these devices are sensing capable DBS devices, meaning we can sense neural activity while we're stimulating the brain. The Summit RC plus S is kind of an improved version where we're capable of doing time domain local field potential recordings at home. So I'll focus on the second 3 participants implanted with that device. So across these 5 participants, there's some overlap in principal OCD symptoms, but the symptoms really manifest in different ways across each individual. And I also want to note that all 5 of these patients are responders to DBS. So during DBS surgery, the deepest contact on each lead was placed so that it's touching either the bed nucleus of the stria terminalis, the BNST, or the ventral striatum. And that's based on awake intraoperative testing. So here I'm showing P4 as an example where the tip of the right lead was targeted to the bed nucleus of the stria terminalis, which is just posterior to the anterior commissure in orange. And the tip of the left lead was targeted to the ventral striatum, which is just anterior to the anterior commissure. The stimulation and sensing contacts for this patient are indicated by the color of each of the 4 spheres here. So red is the stimulation contact. We sense in a bipolar configuration around the stimulation contact to reduce artifact. So those are the green contacts, and the black contact wasn't used. So really we're sensing with one of the sensing contacts kind of anchored in the gray matter structure, where the top sensing contact is solidly in the white matter anterior limb of the internal capsule. So as I mentioned, we do intraoperative awake behavioral testing to determine whether the BNST or the VCVS is optimal. And what we look for during the behavioral testing is a mirth response, positive affect and smiling, or other positive behavioral effects during acute stimulation. And so I'm going to show you some example videos of the positive behavioral response in the operating room. So this patient was left VCVS and right BNST. This is the patient from the image I showed you on the last slide.
Whoa, okay, yeah, I mean, like an 11 out on a scale of 1 to 10, that was something.
It's good though, right?
Yes, something very good.
What do you feel like doing?
jumping out of an airplane.
With a parachute?
With a parachute, yes.
Okay, my next example is dual BNST.
What do you feel in your chest?
Love.
What do you feel? I'm sorry?
Love.
Love.
Uh-huh.
Wow.
Towards you or from you to others?
From me to others.
Uh-huh.
And here is another example of dual BNST.
Remember that. You're gonna have it again. Yeah.
So the OR experience is always like a really kind of like emotional and exciting one. And we see this response in the OR. We usually see it on the same stimulation contact after stimulation is first turned on during initial programming, but this response does kind of wane over time after stimulation. So after surgery, the participants return biweekly to monthly for DBS programming adjustments and behavioral and electrophysiological testing as a part of our study. And so here I'm showing clinical outcomes for P4, that first participant that said he wanted to jump out of a plane with a parachute, over weeks since DBS on. And here in blue and green, I'm showing the Y box and Y box 2, hypomania with the Young's Mania Rating Scale in pink, and depression with the Hamilton Depression Scale in orange. And the participant met responder status at the point labeled with a star at 30 weeks. So we have an idea of how this participant's doing when they come into the clinic at about a monthly basis. But one big question that we need to answer in order to find a robust biomarker is, how is this participant doing in between clinical visits? And how are the symptoms changing on a daily or moment-to-moment basis in natural environments? So this what's going on in between visits question is really what we're trying to get at with this work. So to get at that, we're using the Medtronic Summit RC Possess device to stream intracranial LFP both in the clinic and at home, and we're able to synchronize neural data with behavior. So in the clinic, we estimate changes in affect in response to changing stimulation parameters over short timescales by estimating positive affect via automatic social affect recognition, or AFR. And at home, we have participants record in a wide range of scenarios daily, during exposure and response prevention therapy, during sleep, during behavioral tasks, and during multi-day marathon recording, so that we can capture neural activity at all different times of the day. So participants control their own recordings at home by using a patient-facing application developed by researchers and software engineers at Brown. Participants can launch behavioral tasks from the same application, and there are automatic impedance checks at the start and end of each recording. Each time the participants do a recording, we ask them to rate their intensity of OCD symptoms using the Rune Labs mobile application, which is tailored for OCD. And in this talk, I'll be focusing on the self-reported intensity ratings, which they use this 0 to 10 slider bar to submit. But the participants are also logging information about their mood, sleep, and food intake, and medication intake. So to give you an idea of what the first programming session is like, I'll show another video, this time participant P5. So this was the third OR video, the last OR video that I showed. So here we're recording heart rate through ECG and an optical sensor, 64-channel EEG, LFP and acceleration from RC+S, along with DBS amplitude adjustments. So this video was used to estimate positive valence on a 0 to 5 scale using 2 facial action units related to the cheek raising and lip stretching that happens during a smile. And so as you watch the video, look for positive affect right here to increase and decrease with changes in DBS amplitude. here.
That's weird. Weird. You guys are shaking my body. It's odd. It's weird. My inside's not shaking.
We're back to where we were.
Okay, it's not shaking. How weird. It would literally, when you turned it off, I didn't even realize it, but it went back to doing this on it. I didn't even know I was doing that.
Yeah.
It's not doing that right now. Oh my gosh. It's like the best thing ever. That's cool. Cool. Weird. It's not, like I'm not internally trembling, like fear-wise. Oh, it's different. Cool. That's probably the biggest thing I can feel different, is there's not like a, other than just feeling like a breeze feeling, but other than that, those are the two
Why are
things.
you tearing up?
Because it feels nice that I'm not shaking, and I didn't even know I was shaking. Like it feels, it's a good thing. Like I didn't realize it till you turned it off, and it like, my stomach inside felt like it was doing this. And I was like, oh, am I more anxious again? Like, why is that? Wow. Okay. Guys, this is so freaking cool.
Yeah.
Holy crap.
So as you saw, this patient described kind of an internal tremor in her chest and kind of stomach that she didn't even know is happening. But when the DBS turned on, she realized that that tremor kind of went away, which we thought was really interesting and exciting. So this was like a really emotional, happy moment for this patient. And this was the first time she felt relief in a really long time. And you can actually see this emotion getting picked up in the data. So we're estimating positive affect intensity based on the video in response to changing stimulation parameters. And we saw a peak in positive affect during the portion of the programming session when stimulation on the left hemisphere increased from 0 to 4.5 milliamps in steps of 0.5. And the right hemisphere during this whole period was actually off, not stimulating. So in the future, we're excited to use this data to predict, well, to see if it's possible to predict positive affect based on neural data. At home, we're monitoring symptoms in between visits and over longer timescales. So these plots show the self-reported intensity of OCD symptoms that the patients are reporting through the Rune app on a scale of 0 to 10. So they use their iPhone to do this. And from top to bottom, we have participant P3, P4, and P5. The colored points are the symptom intensity ratings, and the solid line shows the Y-box2 score. Higher Y-box2 means more severe OCD. And as I mentioned, all three participants are responders, and the time point at which responder status is met is indicated by these stars. So after responder status is met, there's really little variation in OCD symptom intensity for the top patient, P3, and the bottom patient, P5. However, P4 in the middle, who reached responder status at 30 weeks, continues to have really wide variation in symptom severity over days. And we find this variation really interesting because it shows that even after responder criteria is met, there are still like good days and bad days. Symptoms are really fluctuating. And this point acts as further motivation for developing an adaptive DBS system that can respond to these fluctuations in symptoms. So each time the participants do a recording at home, we can associate each recording with a particular symptom level. And this bar chart summarizes the amount of data we've collected at each symptom intensity level, along with how much data we've collected during behavioral tasks, exposure and response prevention therapy, and in total. So in total, we've collected over 1,000 hours of LFP across the three participants at the time this was published. And notice that the majority of data is associated with symptom intensity below 3 out of 10. So for P4 in this teal color, he conducted the most hours of recordings at home, and we found that he was most likely to engage in recordings when he was feeling well. So this reporting bias makes it difficult for us to identify trends in neural data related to symptom intensity because we don't have a lot of data collected during elevated symptom states. So one way we're trying to tackle this reporting bias is by conducting multi-day marathon recordings where the patient's asked to record for over 24 hours. And here I'm showing an example marathon recording with participant P4, where he recorded for over 30 consecutive hours with breaks for recharging. So the participant recorded heart rate and acceleration on Apple Watch, LFP and acceleration on the Summit R/S, and he submitted OCD intensity ratings on about an hourly basis. And the 90 seconds of data that I'm showing here just show an example of these streams. And he submitted an OCD symptom intensity rating at this time point. So P4, this participant did one 3-day-long marathon recording where there were natural exposures to OCD triggers on days 1 and 2, which we found was pretty rare to find in one of these recordings. And this recording stood out as a great opportunity for analysis because we don't often see such wide fluctuation in logged OCD symptoms within the same recording. So our goal is to develop a classifier that distinguishes between low symptoms and high symptoms. And in order to do that, we need to collect a lot of data associated with both of these states. So LFP availability over the 80 hours here is shown by the orange shading, and self-reported intensity of OCD symptoms is shown on the y-axis. So to analyze the local field potential data, I computed spectral power in frequency bands of interest for the 2-minute segment of data collected around each symptom intensity rating. So if there is a change in neural activity in the ventral striatum or VNST during symptom provocations, we may be able to pick that up in spectral power of LFP. So here I'm showing spectral power versus symptom intensity for delta, theta, alpha, beta, and gamma for both the left and right VCVS. And I found that VCVS spectral power shows correlations with OCD symptom intensity, where the correlation between symptoms and delta band power showed the strongest correlation, and beta, alpha, beta, and gamma band power showed relatively weaker correlations. And we're really excited to see this because this was our first piece of evidence that VCVS activity might reflect OCD symptoms acutely. So another opportunity to record neural data during OCD exposures and variable symptom intensities is during exposure and response prevention in therapy. So the exposure in ERP refers to exposing yourself to thoughts, images, objects, and situations that make you anxious or start your obsessions. And the response prevention part refers to making a choice not to do compulsive behavior once the anxiety or obsessions have been triggered. And all of this is done under the guidance of therapists. So during the 2-month cognitive behavioral therapy booster phase, participants do about 13 ERP sessions. And this chart shows the data available for each stream during each session over the 2 months. And I boxed out this particular session where the fear trigger was a wart on the hand. And this plot shows heart rate collected over the whole session. And throughout the session, the participant verbally rates their subjective units of distress or SUDS ratings. And we have annotated that from the video and synchronized it to neural activity and heart rate. So as you can see, the participant's SUDS rating was elevated for over 25 minutes after the exposure was introduced. And he started at a 7.5. The exposure was introduced at this vertical black line. So we usually see that participants have some anticipatory anxiety associated with the session. So the participant in the video, again, P4, has very severe OCD specific to contamination. The thought of having a wart is really distressing. And so the image helps to facilitate thoughts about having a wart. He identified thinking about warts as a rather difficult exposure and had to complete a number of contamination-related exposures to build up to this. And he worked through his hierarchy, and after enough practice, thinking about and looking at images of a wart was within his ability to tolerate.
I'm recording on the watch as well.
Can you see this okay?
Yeah.
Okay, so right here is where we're talking.
Yeah, I see it.
0 to 10?
8.5. Oh gosh. And now I'm looking at my own hands
That's good.
for one of those.
So your OCD is concerned that you could have one right now?
Yeah.
Okay. This is really good work. I know that this was a huge trigger.
Yeah, it is.
Even that you're willing to look at this, to purposely look at this, like that's awesome. Let's keep rolling with this. So what are we doing?
Okay.
Why is this wrong and dangerous? And how are we just gonna resist those urges? We're not correcting this.
Yeah. Okay. You can still see me, right?
Yep.
Huh? Yeah. Okay.
Yeah, I can.
Ah.
Suds?
8.5. Still very strong 8.5. Oh god. I'm recording on.
So over all the ERP sessions across all 3 participants, we saw a good range of subjective units of distress, or SUDs over each session. And so for this reason, LFP recordings collected during ERP teletherapy present another unique opportunity to capture neural data during fluctuations in symptom severity. So as an example, I'll show results from analysis similar to the analysis of the marathon recording that I already showed from this boxed out session here. So here, the SUDS ratings ranged from 3 to 7.5 throughout the ERP session, and the vertical black lines show the times of each exposure. And as you can see, the time course of the SUDS ratings shows some habituation after each exposure. So I analyzed the LFP data in the same way as before by computing spectral power in our frequency bands of interest for the 2-minute segment around each symptom intensity rating. And here you can also see some interesting correlations between SUDS and spectral power across these different frequency bands. And while delta band power didn't show the strongest correlation here across all the bands, we did find that the relationship between delta band power and symptoms was preserved across the planned exposures during ERP and the natural exposures during the sprint recording in this patient. So the recording platform I've described enables this type of analysis. I think we have a lot more work to do to nail down a biomarker and be really confident about delta band in this participant. But future work will determine the degree of consistency of these brain behavior relationships across patients and their potential as biomarkers for adaptive DBS applications. So before I talk about our next steps, I want to summarize the significance of this work. So for the first time in patients with psychiatric disorder, we demonstrate the utility of at-home LFP recordings. We captured over 1,000 hours of LFP data at home across various symptom levels and during ERP and DBS. We demonstrate the ability to measure VCVS spectral power during natural OCD symptom provocations at home. We identified decreased delta band power as a candidate biomarker of high intensity OCD symptoms. And continued opportunities for long-term naturalistic intracranial electrophysiological recordings will propel biomarker discovery for OCD and other psychiatric disorders. So next, in the next 10 minutes or so, I'll talk about the emerging challenges that I think we face next as a field. So first, we need to identify robust behavioral and electrophysiological biomarkers. The utility of self-report assessments are limited as they rely on patient insight into their own psychological state, which can be variable. So we really need passive behavioral monitoring. So not SUDS ratings, but instead maybe with wearables that track exercise and activity level. I think that might provide a more objective, accurate measure of symptom state for psychiatric disorders. I also think we need to automate electrophysiological data collection to capture data at all different times of day during different mental states and decrease the high burden that we put on participants during this study. Until LFP recordings can be triggered without participant input, I think they'll be subject to the participants' motivation, anxiety, and mood, which then makes it difficult for us to collect enough data across our good symptom state versus bad symptom state to develop our classifiers. Next, I think we need to harness network connectivity estimates. So obsessive compulsive behaviors implicate symptomatic brain networks. And we really need to increase the coverage of our approach to a few key regions of the corticostriatal reward network, such as the orbitofrontal cortex or anterior cingulate cortex. Dysfunctional reward processing, such as hypersensitization or desensitization to reward, is a common feature of psychiatric disorders. So by looking at what's going on in this reward network, we might be able to improve our chances of finding a robust biomarker for psychiatric disorders. So in phase 2 of our study, we added 2 additional sensing electrodes in the OFC, in addition to the sensing and stimulating electrodes in the VCVS. So these are passive sensing electrodes in the orbitofrontal cortex that will allow us to look at communication between the ventral striatum and the OFC. So there's been some great work by others on reward processing focused on VCVS. In a study of 18 participants with OCD and 19 healthy controls, Viggy et al. Found that OCD patients showed attenuated nucleus accumbens activation during reward anticipation. Lega et al. In an N of 1 with treatment-resistant depression conducted intraoperative recordings and found that nucleus accumbens alpha band power increased during positive feedback trials, and trial type modulated the phase of alpha-gamma cross-frequency coupling. And for positive feedback trials, gamma was elevated during the peak of the alpha oscillation. During negative feedback trials, gamma was elevated during the the trough of the oscillation. And then in 2017, Wu et al. Found increased delta band power in mice and 1 human during reward anticipation. In mice, they were able to use delta band power as a biomarker to control responsive neurostimulation in a way that blocked binge eating in mice. And lastly, Miller et al. Recently characterized dominant oscillations at various locations above the nucleus accumbens in white matter, and found that as they moved down the anterior limb, theta, alpha, and gamma oscillations were present at different depths. They also found that provocation of OCD symptoms modulated gamma oscillation amplitude within the nucleus accumbens. So there are several frequency bands popping out here, delta, theta, alpha, and gamma, and I think it's still up in the air as to what activity in these different bands definitively means, and how VCVS is functioning as one node in this network of reward. So the question that I would like to end on is, could the work we do in VCVS, DBS for OCD help us better understand electrophysiological signatures of other psychiatric disorders? And if we think of reward processing as a functional domain that's impaired in OCD, then biomarkers of this dysfunction might be relevant for other disorders. For example, if we can better understand hypomania, maybe we can better understand the manic component of bipolar disorder. Patients with OCD represent the most number of patients implanted in the VCVS for DBS. So what we learned from OCD, I think, will help us understand other psychiatric and substance use disorders as well. So with that, I want to acknowledge my mentor, Samir Sheff, and the PI of the study, Wayne Goodman, along with our partners and collaborators. And thank you for your attention.
Thank you, Dr. Nicole. That was such a brilliant, fascinating talk.